Purdue University. She's an assistant professor in agricultural and biological engineering department. She is also the founder and CEO of a blazing fast cloud computing company, KeyByte, since 2021. She leads the group called Innovatory of Cells and Neural Machines at Purdue. She finished her PhD in biomedical engineering at Purdue, winning Chorofa's Dissertation Award and the Future Faculty Fellowship. Next, she followed this with a postdoc at UT Austin and a visiting assistant professor stint in Purdue Computer Science. She has published in top applied machine learning, computer systems, computational genomics conferences and journals. She has received extensive funding from NIH, R01, DoD, from Air Force Research, NSF, multiple grants, including a career, NIFA, as well as private industry funding, including Amazon, Microsoft, and Adobe Research. She serves on technical program conferences in IoT, computer systems, machine learning, computational genomics, and recently selected to participate in 2023 Japan America Frontiers of Engineering Symposium in July in Tokyo. The floor is yours, Professor.
Thanks, Mohammed. Thank you, everyone. Thank you for abstracting the really long title I had for my talk. So fast, furious, and interpretable machine learning. And I did manage to squeeze in IOST. So I will try to mostly focus on genomics because that has a direct mapping with your institute's mission. But as I would have it, I did manage to also put in some facets from my NSF Career Award. So IOST, for those of you who are not familiar, it stands for Internet of Small Things. So that's my take on Internet of Things, where I wanted to emphasize smallness. And then at the end, I will end with a little bit about my company. It's a cloud computing company called KeyByte. So the vignette that you saw in the tweet comes from Vignette 1. So I will focus on Vignette 1 in my talk, and then I will try to give you a teaser for Vignette 2. And then I'll end with my the mission and the victories of my company. All of this work is published, so you will be able to follow anything you want to pick up from my website. So feel free to look at it, look at the code, play with it, and you'll get back with questions. So a little bit about me. I'm the CEO and co-founder of KeyByte. I'm an assistant professor. I specialize in data engineering. I lead the Innovatory for Cells and Neural Machines. I really love the acronym that we came up with, ICANN, at Purdue. We are an applied machine learning lab, and we innovate at the nexus of machine learning and mobile systems. So think IoT, cloud computing on the one hand, and machine learning for health and vitality on the other. More specifically, computational genomics. And thanks to having these two thrusts, I call thrust 1 computer vision and mobile systems, and thrust 2 machine learning and genomics. We have been fortunate to get grants from NIH, Department of Defense, more recently my NSF Career Award, and also private funding from Amazon, Microsoft Research, and Adobe Research. I'm also pretty excited that I'm going to be attending the NE Frontier Symposium this year. So with that, let's dive in. This is a plug. So if any of you are excited enough, we do a lot of different things, but it's, like I said, essentially an applied machine learning lab. Feel free to shoot me an email, and I'll make sure you get a very well-grounded mentorship from Purdue. A lot of things are happening here, including new leadership. So I'm very excited to be in a happening place. So if you want to leave California and come to the Midwest, feel free to holler. Okay, so I will start with Kratos. Kratos is an algorithm that we published last year in ACM KDD, which is a top machine learning data mining conference, to be specific. And the motivation for Kratos is that sequencing is cheaper and much higher quality than was just a few years ago. And specifically for Kratos, we have applied machine learning to single cell sequencing data because I'm really excited about single-cell sequencing, the resolution at which you can map the sequence, the state of a single cell, as opposed to averaging across a population of cells, is what is now doable, both because of the instrumentation and because of the computing that can analyze and compute the downstream stuff from the sequence data reads. So just to tell you why single-cell sequencing data is very helpful and why I am excited about it, and what is the data science, data engineering challenge to go with single-cell sequencing data. So on the left, you see the more conventional sequencing where you have average gene expression from all cells. Now, averaging does not do justice to this. So if you see on the right is the actual vignette, where each cell type has a distinct gene expression profile. And then when you take the average, you're basically smushing it into an average. So you're basically losing a lot of signal. So we don't want to do that. So if at the data generation stage, you're already smushing the signal, well, it's hard to do much afterward. So single cell sequencing really opens up the floor for looking into a single cell at a single cell granularity. And just to tell you what it looks like, you ultimately, after you have the single cell sequence data, the data can be clustered into multiple cell types. And these multiple cell types, you see clusters here. This is something called UMAP, which is used for dimensionality reduction, more specifically for visualization. So you see these clusters are clusters of different cell types. So I want you to keep this in mind so you understand Kratos. And these are different dimensions. So you have different dimensions of the cell type analysis that has been done. So one, I told you, is when you're doing bulk profiling, you're losing signal. And the second, you might actually lose rare events, right? So single cell sequencing data lets us look into cells at a single cell granularity that was not possible about a decade ago. It enables transcriptome-wide gene expression measurement at a single cell granularity. And so you can see these clusters of cells like I showed you in the previous slide, and you can get a much clearer idea of these cell types, both at the spatial level and at the temporal level. We call this pseudo time. So the whole perspective for a cell changes. So now you have this fuzzy state of a cell in a continuum. And based on what you're doing to the cell, right, it's kind of empowering. Based on what you're doing to the cell can be in multiple phases within this fuzzy continuum. So the single cell analysis and single cell sequencing to start with, and then the computation that results in single cell analysis gives you the ability to think of a cell as a continuum rather than these discrete cell types and organs and tissues. And then I encourage you to read this paper from Genome Biology, where they mapped out some of these grand challenges in single cell data science. So of course, we are standing on the shoulders of these giants. We did not come up with all of this on our own. So one of the things that we have to do in a lot of these machine learning conferences, which is the prime venue for results that we get that can beat the state of the art, I will call state of the art SOTA, state of the art algorithms. So oftentimes what you have to do is you have to say, what are your contributions and why are you beating the state of the art, and by how much are you beating the state of the art, right? So I will start on this sort of competitive note, which is kind of the trend of this community, where you identify what are the state of the art algorithms, and then what are the features or the modules in your algorithm that results in you beating the state of the art. So our algorithm is Kratos. And in Kratos, what we do is I showed you the cell clusters, and then you can also do trajectory mapping, right? So if you think of, again, I'm going to try to connect this to the audience. So if you think of cell differentiation, for example, right? So in the past, I've written proposals, and I think including with Ali and his team, where you have cells differentiating from the embryonic cell type or the induced pluripotent cell type to differentiated cell types. So using your algorithms, you can also do this trajectory mapping, cell trajectory mapping along the pseudo time. So that is what I mean by trajectory. So you have these intermediate cell states. And the exciting thing about single cell genomics is then you don't have to think of these discrete, sort of more conventional ways of thinking of cells, where you have these very static organs, static tissues, etc. But you have the cell that is now empowered to, depending on what you're doing to the cell, the ambient conditions of the cell can be in this oscillation across multiple different states. And that's what single cell genomics allows us to compute. So the benefits by now, just to recapitulate, it measures gene expression at the individual cell level. For today's talk, I'm going to focus on gene expression, but you can also have multiple additional features from an ML standpoint. You could have epigenetic features that we have worked on in the past. But this is funny, like I think you had Peter Diamandis for a talk, and he writes a book every 5 or 10 years. So it's funny, every 5 years, my slide deck totally changes. So 5 years ago, I would talk about epigenetic stuff, but now since we're moving on to single cell genomics data, now we are starting off with gene expression. But of course, you can increase the dimensions and look at additional epigenetic features. But like I said, for today's talk, we will consider gene expression in the matrices. So if you think of a single cell genomics matrix, right? If you think of a matrix, you have rows and you have columns. Now, the rows of the matrix, remember this, are different cell types, and the columns of the matrix are different features. In this case, these are gene expression levels, right? So you have, this is called a count matrix, and the count matrix is what is basically the raw data that goes into Kratos and other such algorithms. Now, what's the challenges, right? So I'm going to kind of blend data science terminology here. One is high dimensionality of the data set. So typically what we mean by high dimensionality is when you have many more columns than the number of rows. And the more the number of features, the more is the curse of this high dimensionality. The other thing is sparse matrices. So there are a lot of zeros in the matrix, which kind of makes it another challenge from a data science angle. And again, there are limitations of current sequencing technologies. So the data is noisy. And some of the identified challenges from, again, a data science perspective, because we are looking at this from a data science angle, is one, there is batch correction. And this, I'm sure all of you are very familiar with. If you have different labs generating data, you will have artifacts of those different labs generating data, so different batches of data, or, so that is that spatial batch effect, or you could have temporal, right? So if you're generating data over time, just because of generating data over time, maybe using different protocols, you will have these batch effects. So that is something that we have to deal with. The other thing that we have to deal with is something called a dropout event. So what happens in a dropout event is you actually have a signal, but the instrumentation is not resolved enough to be able to pick up that signal. So these are two of the initial challenges that we are focusing on, and that Kratos, our algorithm, takes care of. So one is batch effect, the other is the dropout event. And as a result of this, the data might look different or deviate from the original data manifold, right? So these are some of the challenges in single cell sequencing data analysis. So if you look at the top right, you will see some of this pictorially. So you have an observed data point. So remember this, when you have an observed data point, that is the ground truth data point. And what you're predicting as a result of your computation is the predicted data point. So a lot of things that we have to do, and which you will be familiar with, if you want to collaborate in the future, is, for example, denoising. Right? So we have denoising algorithms. We can impute data points that were not experimentally gathered. And so we are able to do denoising, we are able to do imputation. So we are able to augment the data manifold, and we are also able to take care of the deviation from the original data manifold using our algorithms such as Kratos. So here on the top left, you see Charles DeLisi, who got some of this, who was responsible for the congressional funding for all the genomics revolution that started happening with the genome project, Human Genome Project. And, so that was awesome. And then now we have moved on to, much higher resolutions. Now we are talking about single cell. So there you have the whole genome, and now you have the single cell data. Now, some of the data engineering or data science challenges, I do both data engineering and data science, and hopefully by the end of this talk, I would have made some, the boundaries are again fuzzy, but hopefully you would have taken away something from today's talk. So some of the things that we want to do is we want to cluster. So like I showed you in one of the earlier slides, you have clustering. So you have different cell types, and you cluster these different cell types based on things such as gene expression enrichment, right? So if you have a gene panel, rather than having a single gene that is going to identify a cluster, you have a panel of marker genes. So this is another thing that the field has moved toward, that a cell type is not characterized by a single gene, but by a panel or a vector of genes where the genes may be upregulated or downregulated. So it could be the presence of the genes, the increased expression of the genes, the decreased expression of the genes, or the absence of the genes, right? So it is more of a marker panel rather than a single gene. And then the other thing that we are also excited about, which I think is very relevant to the National Institutes of Health's mission, is to look at the trajectories through cell space, right? Think of the cell as a continuum, that the cell can be empowered through space and through pseudo-time. And then comparison of multiple methods. So there are different methods that take care of different aspects. So Kratos, for example, takes care of the dropout event that we talked about. And we are also developing techniques for batch correction, which again, I think is a very practical problem in this domain, both bioengineering and computational genomics. Now, a cell's identity, just to kind of hit on that a little more, you can read some of these papers here, but a cell participates simultaneously in multiple biological contexts. And this is what I think connects computational genomics to a lot of the work that is going on in your institute, that depending on the environmental stimuli, you could have these different the cell being kind of coexisting in this continuum of spaces, right? So think of the time continuum over time, depending on, healthy stimuli or unhealthy stimuli maladies, and think of space, right? Where the cell differentiates spatially to form organs and tissues. So you have this continuum of cell identities, and we like to think of this as basis vectors that map a cell to a continuum of cell identities. So one of the things that we want to do is, this is what you get as a result of single cell genomics data that goes into our algorithm. So I talked about this multi-dimensional data. So the columns, they are the genes, and the rows, these are the different cell types. So our question that we are asking is, what are the genes that are typifying these different cell types, right? So here on the bottom right, you'll see cell type A, cell type B, cell type C, and so on. So we want to be able to cluster these cell types. And the other nice thing is, as a result of clustering these cell types using this high resolution data, you can come up with new clusters that no one has found before, right? And maybe the cluster is an important cluster. So that's where I think there is this loop between data scientists and experimentalists, where you can go and validate that new cluster and say, hey, maybe this cluster has this new gene panel that is enriched in gene A, gene D, and gene Z, with these mappings between them. So we found this new cluster, which is possibly important for Alzheimer's disease, say. So this is kind of to give you an idea of why we developed Kratos. So in Kratos, again, to get a little bit into the machine learning side, on the top right is Zihan Zhao, who's the lead developer for Kratos. What we do is, the first step is dimensionality reduction. The second step is cell clustering. And the third step, which is very important to application scientists, is interpretability. Right? So we want these explanatory gene panels that typify those different clusters. So the first step is gene dimensionality reduction. So just to give you an idea, PCA, for example, principal component analysis, where you find the eigen values for the eigenvectors. So it's kind of, you're finding the important dimensions that are representing a specific type. So that is how we typically do a gene dimensionality reduction. And then you do cell clustering. So one of the things that we do is, again, this is from the data science perspective, if you can optimize two steps together rather than doing each of these three canonical steps separately, you would basically, through this joint optimization, get a better performance. And hopefully, by the end of this, and you can read the paper, you'll see that one of the reasons we have better performance than the state of the art, remember I called it SOTA, is that we combine the gene dimensionality reduction step and the cell clustering step into a joint optimization. So we use a neural network in order to combine gene dimensionality reduction and cell clustering. As a result of that, we get much better clusters. One, the performance of the clustering improves, and second, the interpretability of the clustering improves. So we are able to get clusters that with higher confidence we can match to different cell types, which is again very important to this domain. So again, these are the different cell types, and these are the class assignment scores for the different cell types. So here, to tell you now, so I show you some of the results. This was the state of the art before this. So ICML is another top conference. So in computer science, we primarily publish in very highly peer-reviewed selected conferences. So ACE was from ICML 2021. And if you see our clustering is much better resolved. So we are able to perform better. And then we are also able to interpret the clusters a lot better by using this joint optimization. So Kratos is our method. ACE is the baseline method that was best before Kratos. So what we do in Kratos is we combine dimensionality reduction and clustering into a classification neural network to avoid separate optimization. So in a sense, clustering is an unsupervised machine learning task where you come up with these clusters. We are converting that to a supervised machine learning task using high confidence labeled data sets. And a supervised task is generally much easier than an unsupervised task. And as a result of that, we're getting better clusters. And then again, I will just go ahead with this. If you have questions, you can come back later. We use something called perturbation. So perturbation has been used in computer security literature, where you perturb a model so that the model gives you something different from what the model should have given you, right? So the right result of a model prediction, say, is a cat, right? So we perturb the model so that instead of giving the output as a cat, it gives the output as a dog. Right? So that is what a perturbation is in the machine learning parlance. Now, what we do is we use perturbation for explanation, right? So we're kind of giving a twist to perturbation and use it for explanation. So the way we do it is we see that the gene expression, so depending on how much perturbation the gene expressions require for the class assignment to change, so the more the perturbation that is needed, the more important is that gene or that group of genes for the final assignment. So to give you an example here, you have this neural network. So I said we are combining steps 1 and 2 using a neural network, which is giving you this class assignment. So these are the different classes. Now we do perturbation. So we are perturbing the gene expression. And as a result of that, we get a different class assignment, right? And depending on how much you have to perturb a gene, that gives you the explanation or the importance score of that gene. So we see that the ability to also explain these gene panels that are typifying the different clusters is also higher, quantitatively profiled when we use Kratos. So I'm going to kind of go through this. So I made my point. This is the architecture of the neural network that we are using. It's a simple neural network. If you remember in my talk today, I said fast, furious, and interpretable. So oftentimes we don't want something super complex, so we can interpret the results. So this is a simple neural network that we used. And the perturbation is something that is, again, used commonly in computer security literature. This is called the Carlini-Wagner perturbation, where you want to make sure the perturbation is sort of stealthy. So you're learning this perturbation function. So the perturbation is good enough, but not loud enough that the bad guys or the good guys can see that you're perturbing the model, right? So remember, I took this from computer security literature. And in computer security, you kind of want to make sure when you're perturbing, when you have an attack, the attack is stealthy, right? So you kind of optimize this perturbation function so that it's good enough, but not loud enough to be able to be caught by the good guys, right? Okay, and then so what we do is we evaluate. So a big thing in this domain is you have to evaluate. So you need baselines. One baseline I told you is ACE. We used another baseline called GCE. All of this is available on my website. The other thing is data sets. So a lot of this, you need benchmark data sets. And the advantage of working with, big lab is that, not only would we have these data sets that are out there, but we'll be able to validate maybe some unknown cell types using new experimental data, which is kind of determined by the prediction of my algorithm. So kind of going back to my original point is we have a combination of real data sets. So we use 2 real data sets from 2 Nature papers, recent ones. I have those citations in a bit. We call them the PBMC data set and a more complex data set called the Barron data set. So again, when you have data sets, we kind of stress test or challenge our algorithm using more complex data sets. So Barron is the more complex data set, and PBMC is the relatively simpler data set. And then we also like using simulated data sets. So we use something called SimSen. which is a package where we know the ground truth data, right? So we can really benchmark our algorithm using ground truth data. So there are three things to remember here, so you understand my results. One is you have causal genes, which are the important genes. The other is the dependent genes, which are kind of related to the causal genes. And the noise genes are kind of the housekeeping genes, right? So in this context, they're no good, right? So causal genes, dependent genes, and you have the noise genes. And so these are the three things that we have to simulate using the SimSim, and we did. And then as a result of this, I'm kind of going to, this is a sort of a digression, but maybe we can talk about it later. But one of the things for those of you who are data scientists is this falls under latent space learning, because you're kind of trying to learn the latent data manifold, which kind of removes all the noise, right? So I'm going to kind of just ignore this digression, but kind of wanted to bring this home, that a lot of the work that we are doing is latent space learning, that in that latent space, if you're projecting from the original data space, you get a lot less noise. So we can learn various things in the latent space. And something that might actually be helpful for you guys is in the latent space. For example, if you have a drug treatment, or you have some kind of maybe a mechanical treatment to the cells. So in that latent space, you're able to model that effect better. And then if you put it back to the original data space, you can basically get the mapping. So since I brought this up, let me try to explain this a little bit, and then we can move on. So there is something called an encoder and a decoder, right? So an encoder and decoder structure, and this might be helpful for latent space learning. And if there are data scientists out there, that's used very popularly in machine learning. So kind of to go through encoders and decoders, the reason I bring this up is the encoder encodes into a latent space. So you get an embedding. So if this is your original raw data, in the embedding, you remove a lot of the noise by abstracting the important features in that embedding. So the embedding is almost like a bottleneck layer that preserves the important features, right? So you're kind of throwing out all the noise, and you're kind of gathering the essence of the data. So one way I like telling students is that in the embedding space, if you think of a photocopier, you're making a photocopy, but the photocopy does not preserve all the unnecessary details. It captures the essence of the space. And then basically, if you copy it back, the decoder is almost like a sanity check. So I take that a little bit, I'll backtrack a little bit. So if you make a copy, you have the encoding and the decoding. The closer the decoding is to the original data, the better is your embedding, right? So the decoder is almost like a sanity check. So if the decoder gives you data that is very similar to what you originally had, that means your latent space learning is more appropriate, right? So this is sort of a digression, but hopefully for those of you, who want to do this, this has got, we're working on this to kind of denoise the single cell genomics data and learn stuff in the latent space. So for example, if you have treated cells and untreated cells, we are able to do a lot of this perturbation and learning in the latent space rather than in the original data space, which is a lot noisier. Okay, so coming back to our trajectory, so we have SimSim, which is generating a synthetic model. I showed you how we are generating the model, where we have the causal, the dependent, and the noise genes. And then we have two real single-cell RNA-seq data sets, the PBMC, which is slightly easier. You see that the cells and genes and the cell types. The reason I say this is slightly easier is because the dimensionality is a little bit lower, so it's easier for the machine learning algorithm. The second is the barren data set. And then we compare the visualizations of that latent space. If you remember from the previous slide was my quick blurb on latent spaces. And then our baselines are two top algorithms from ICML 2020 and ICML 2021. And then we also generalize. So we show that our algorithm does not just work for genomics data, but it also works on an entirely different domain, computer vision. So MNIST is a data set which is commonly used in the computer vision domain for image recognition. So it also works on an entirely different domain. So clustering metrics, and again, there are students in the talk, these are commonly used clustering metrics. One is adjusted mutual information. Another is adjusted random index. Now, ARI works slightly less well. So AMI, let me put it this way, AMI works better when there is class imbalance. And silhouette score basically shows you what is the shape of the different clusters. So it's a little more cosmetic than the AMI and the ARI index. And the higher the AMI or the higher the ARI, the better is the clustering. Whereas the silhouette score, again, the higher the silhouette score, the better is the shape of the clusters. So if you remember, I showed you the shape of the clusters as a result of Kratos versus ACE. Now, AU-ROC is used again as a quantitative measure. So higher the discriminative power of your top important genes, the higher the area under the ROC curve. That's what AU-ROC stands for. And higher score meaning more representative top genes. The Pearson correlation coefficient, I'm going to ignore that right now because it's a little bit getting into the weeds, but it's to see the redundancy of these top genes. And it's kind of an auxiliary metric that we use for Kratos. So this is Kratos versus ACE. So we see that this is, remember, I told you, you have the causal genes, you have the dependent genes, and you have the noise genes. So the dependent genes are an auxiliary metric, but the noise genes are important. So you'll see that in Kratos, the first three are for Kratos and the latter three are for ACE. You'll see that ACE gets a lot higher number of noise genes, which is bad. You want lower number of noise genes, and you want higher number of causal genes. And the dependent genes are kind of okay. So if you have dependent genes that are kind of getting co-regulated alongside the top genes that are typifying the cluster, so it's kind of an auxiliary metric. So what we see is that Kratos beats ACE hands down using all of these three, the causal, the dependent genes, and the noise genes. And then you see that I told you there are three metrics, quantitative metrics, in order to be able to evaluate the performance of the clustering. So one is the ARI. So you see that the ARI is highest for Kratos. Again, AMI, which is for slightly more imbalanced data sets, is again highest for Kratos. And ACE and GCE are the two baseline algorithms, right? The competitors that we beat and we evaluate using the benchmark data sets. So again, you see that we also have this is the raw data, this is using Kratos, and this is using ACE. So we see that Kratos is able to also resolve the clusters a lot better. So the silhouette score is basically more cosmetic. That shows you the shape of the final clusters. So this is one of the data sets. The reference for one of the data sets is from Nature Communications. Thanks to them, we have access to the data set to be able to try out our algorithm. And the other data set is the Barron data set that is from Cell Systems 2016. So these are used as benchmark data sets for single cell genomics clustering. And we see that, again, if you consider the AU-ROC, this is again, I might even skip this, but this is related to the interpretability. So we used three different metrics for interpretability. And Kratos has three variants where we have Karleny-Wagner index, which is the perturbation technique that we use to see the importance score of the different genes. And we also have IG and SG, which is smooth grad and integrated gradient, which are two other interpretability metrics. And we see that the performance using these three different interpretability metrics is different. Like I said, I'm going to skip this. The final thing I have here is oftentimes you want to make sure that your algorithm works outside your target domain. That's called generalization. And we see that algorithm also works for the MNIST data set. What the MNIST data set is, for those of you who are not familiar, is basically handwritten digits from 0 to 9. So here, interpretability, just to give you an idea, if you think of a 3 to 8, right? So if you want to convert 3 to 8, then you basically would have to fill in the top here, and then you would have to fill in the bottom left, right? The top left and the bottom left. So basically what our algorithm tells you is the direction of the change at the pixel level, right? So what do you have to add or what do you have to subtract to perturb one digit to another? Right? So this is to show that our algorithm does not just work for single cell genomics, but it also works for a very popular computer vision data set called MNIST, which is a digit recognition data set. So it will also work for a simple computer vision data set. Okay, so with that, I will quickly switch gears. So hopefully I have told you that we are doing some exciting work in single cell genomics. This can also be applied to different types of clustering, right? So if you have different kinds of drug treatments, if you have different kinds of mechanical treatments, we are able to cluster the cells and tell you what are the different genes that are upregulated that define those clusters, right? So with that, I will actually move a lot faster and tell you some of the additional exciting things that are going on in our lab. So if you guys are interested in nano core reads. We are doing error correction. And I couldn't help myself but pick up a paper from a couple years ago where we've used these language models. If you're familiar with GPT-4, we've used a very simple variant of GPT-4. I'm just going to put it that way, transformers for error correction. So a lot of these data reads, whether short reads or long read sequences, are full of errors. So we are able to do error correction using our algorithm. This is again published, Learner. We are also able to, I'll talk in a little more depth on number 2 and number 3, maybe 10 minutes to 5 minutes each. But just to give you an idea, this was led by Atul Sharma and Pranjal Jain, the auto parameter tuning for error correction. So a common theme in our lab is how do you use machine learning to do auto reconfiguration? Hopefully you'll at least get what I mean by auto reconfiguration by the end of my talk. So to give you an idea of what Lerna is, so Lerna, again, you can go to my website and it's published. So Lerna comes up, there is something called K. So for error correction and for a lot of these sequence analysis, you have to come up with the best value of K. So just to give you an intuition for Lerna, in Lerna, what we do is we use something called a perplexity metric. So the higher the perplexity of a language, so we almost consider genome as a language. The higher the perplexity of the language, the worse it is. So this is how you represent perplexity. So we use perplexity as a fitness measure, and we basically come up with the best value of the K parameter that is used for error correction. And by using perplexity, what we want to do is the search algorithm tries to decrease the perplexity. So if you think of the language, intuitively, the language should be simple. So we use a concept from language modeling called perplexity and apply it to error correction in short reads and long reads. And like I said, this is again published and you can look this up. I'm not going to spend too much time on this. This is from BMC Bioinformatics 2022. And I think it's funny that ChatGPT and everything came out and we have application of the same type of algorithms to being able to get better error correction in genomics reads. Okay, so now I'm going to actually take you to thrust 1 of my lab, and some of this maybe, I might actually go through this sort of fast, but at least you get some idea of what my NSF career project is on. So what I'm really excited about is, can you run these really large algorithms on small devices, right? Instead of running these on big GPU servers, can you run these on small devices, right? The way I can think of this being connected to an institute that works in biomedical engineering and health is that, think of wearables. Can you make a lot of this work on wearables or handheld instruments or ambient sensors, right? So if you have these sensing devices, can you make the sensors more intelligent by being able to do some of the computing on the sensors? And what do we want to do? We want to derive health indicators from all of this data in real time, in a privacy preserving manner, because you're not transporting them to the cloud, and in a cost-effective manner, right? I'm not going to make the analogy now in the interest of time, but so one of the things that we do, for example, if you see these bounding boxes, these are basically identifying that these are different human beings with a certain confidence score, right? So this is called object detection. You're putting a bounding box around these different people, right? So object detection is a computer vision algorithm, and it uses something called convolutional neural networks. So can you make these transformers and convolutional neural networks work on small devices is what the vision for my NSF Career Award is. So in order to do that, you sometimes, again, remember I told you one theme in my lab is to use machine learning to configure the parameters of an algorithm, right? So if you think of this giant neural network, this gas-guzzling neural network, by gas, I mean compute-guzzling neural network, right? So if you basically want them to run on these small devices, you have to reconfigure the parameters of the algorithm and kind of squish it so that it can run on these smaller devices in an energy-efficient manner and can run to give you results in real time, right? That is what we call real-time latency. So you kind of want them to give you timely results, that is with a lower latency, and also be energy efficient, right? So can you adapt these algorithms to run on these small devices by decreasing the parameters of these large algorithms. So that's essentially what we do. And the other smart thing that we do is we are able to do two things. One is make them content aware. So if you have a very complex scene in a video, you need less computation. So we are intelligent to decrease the computation if you have a simple scene, right? So think of just audience sitting in a hall versus think of a soccer game and everyone's running, right? So the second one is a much more complex scene where you'll need more computation. So based on the content, we are able to adapt our algorithm. The other thing is think of your phone, right? So your phone, say if you're driving a Tesla, your phone, you might be checking the weather, and at the same time, it's giving you directions, right? So that is contention. When multiple applications are running on the phone at the same time, that makes it more kind of harder for the phone to do this computation in real time. So it's not just one application, but multiple applications. So you're content aware, and then you're resource contention aware, because the resources of the phone are being used by multiple applications at the same time. So our algorithms, as a result of my NSF Career Award and a lot of the preliminary data, are adaptive so that they can adapt to the content and the contention. So here comes the transformer, literally what's driving ChatGPT 4. So you have these really compute guzzling algorithms, and can you make them run on these small devices? So thanks to the NSF Career Award and Department of Defense, now we have a lot of these devices, these small mobile GPUs, we have drones, we have wearable devices. And so we are basically transforming our algorithms, quite literally, to make them work on these small Arduino boards. So now we're not just a cyber lab, but we are a cyber physical lab. So this work has been done by Ranshu, who's now in NVIDIA. I'm hoping, we can get additional servers and mobile GPUs. We also have the latest mobile GPUs that run Orans. They call NVIDIA Orans. We have them in our lab. Jian Li, he's looking more at the energy side of things. So can you, if I say, hey, you have 5 hours of battery life remaining, can I squeeze that out to get maximum performance? And Punchang is working with LiDAR data. So we're doing 3D computer vision on these small devices. So when I say real time, by real time, typically we mean 30 frames per second, right? So if you think of these frames of a video, 30 frames per second is real time, which translates to 33 to 50 milliseconds per frame. That's the latency, the real-time latency per frame. So, if you want to read this, this is related to my career award, and it's also on the College of Engineering. They have the Medium blog. So I think you'll get some idea of why we need these sensors, not just to sense, but also to compute. And these are the kinds of devices on which we have this Jetson AGX Xavier, Jetson Xavier NX, and Jetson TX2. So what we do here is we evaluate using different capabilities. When I say capabilities, these are compute capabilities. So we change the computational resources of the devices. So if you give me a less computationally resource device, we are able to aggressively approximate our algorithm to be able to run on the type of device that you give me. So kind of bring your own device, and we are able to adjust the knobs of our algorithm. So this work, again, has been published. And, I'm happy to talk offline because I feel that there are possibilities, in wearable sensors, health sensing, IoT, etc., and being able to adapt and approximate algorithms. Not only that, it also makes algorithms less energy hungry. And guess what? It also makes them more interpretable, because now you have simpler algorithms that can run on these devices. So I'll skip this so I can go to the last part of it. I pretty much told you this. We have the content-aware features that can give you the right level of approximation for your algorithm. And again, similar to what we had like ACE, we are able to beat the state-of-the-art. The state-of-the-art here are from CVPR, which is the top computer vision conference and at a scale. So we are able to take adaptive decisions. And not only that, we are able to adapt based on the kinds of real-time latency that you need and the kinds of energy limits that, so if you tell me, hey, I have 4 hours of battery life remaining versus I have 8 hours of battery life remaining, my algorithm can tune according to the energy, it's called SLO, service level objectives, that are set by the client. So both from the timeliness side, how fast you want the thing to go. So think of surgical robots, right? They have to basically see and work at real time. So we wrote a proposal using surgical robots where you have real-time computer vision. And then think of these running on small battery-powered devices where energy is very important. So I'm kind of going to skip through this because, but if you're interested in this, I can talk in greater length. Obviously, there are convex solvers here. One thing to keep in mind here, what we are trying to do is we give you the maximum accuracy. So accuracy in terms of computer vision algorithms is measured using something called MAP, mean average precision. And we are able to adhere to the latency requirements and the energy requirements. If you say that, hey, my algorithm should not be slower than this, it'll give you the maximum accuracy, taking into consideration that latency requirement. So with that, I'm kind of sped through this because I want to come to the last part of my talk. And our algorithm is called light reconfig because we are able to reconfigure light our algorithm at runtime to be able to adapt to the different service level objectives based on energy or based on latency. And we call it light reconfig. Light reconfig has a cost benefit analyzer. So the takeaway from this is the benefit is in terms of accuracy. The higher the accuracy, the better. The cost is the latency. So sometimes if you have to do very complex computation, you'll pay a cost. The cost will be in terms of more compute time or more energy. Right? So we are able to take this into consideration and give you the best accuracy, given the energy constraints and given the latency constraints. So light reconfig, again, this is published from last year, Eurosys. This is on my website. It's a cost and content-aware video object detection system. It's also the basis for my NSF Career Awards. I'm very excited. We are doing additional work on it. It's got a cost-benefit analyzer, so it improves the accuracy, but keeps in mind what are your timeliness and what are your energy constraints, and then it's able to use the state-of-the-art object detection backbone at its heart. The code is available. If any of you are interested, if you see, using variables and computer vision algorithms for things like surgical robots, feel free to give it a spin. Our code is outsourced, and some of this is also patent pending. With that, I move on to the last part, and I'm going to kind of race through this so I have a little bit of time for questions. It's also related to my company. So my company, the vision for my company is cloud computing for all, right? And I know Ali worked in Amazon for a little bit. So I think a lot of the Amazon's AWS, which is Amazon Web Services, a lot of Amazon's revenue comes from cloud computing. We ourselves pay a good cloud computing bill. Thanks to Amazon, we've also gotten a grant. We got a grant last year, so all our cloud computing was sort of free last year, but now we are back to paying the bill. So what KeyByte does is it democratizes cloud computing. So how can you, if you give me, hey, I want to run all my computing for no more than $10,000 per month, then it's going to do these adjustments, reconfiguration, to be able to give you the right kinds of performance for the kinds of costs that you want to see at the end of the month. So the other advantage there, so think of, and hopefully by the end of this talk, you'll at least know this, if nothing else, is think of two sides of the game. And both sides of the game are kind of important. So one side of the game is the Amazons and the Googles and the Microsofts of the world. So we've got funding from Microsoft and we've got funding from Amazon. So they have these data centers, right? So a lot of the times for running all the compute, think of all the ChatGPT, right? It's not free. There is computation that is happening at the back end. So to run all of this, they largely over-provision the number of servers that are running in the data centers. So what our algorithm could do is it could right-size the compute servers that would be needed for your computation. So that is from the provider side, the cloud provider side. Now think of the cloud client, right? The yous and me's of the world or the KeyBytes of the world, right? So from the cloud client side, you have these cost constraints, right? So maybe I don't want to spend more than $20,000 per month for my cloud bill, right? So that is, the cost is more important to the user, and maybe the data center usage, the energy is more important to the vendor, right? So we are able to right-size both the computation, and we are also able to right-size things from the client side so that you're not paying a hefty cloud bill at the end of the month. So that's what KeyByte is about. It's removing all the shackles of cloud computing, so you have crowds cheering at the end, right? That you can get cloud computing democratized for companies of all sizes. So we configure to be able to, again, a lot of this is reconfiguration. So hopefully, again, similar, the common theme here is reconfiguration. So these are databases that are running on the cloud, right? So the databases have their own knobs so that we reconfigure at real time using machine learning. And then we also select the right VM, which is the virtual machine instance that is provisioned to you, right? So we are basically reconfiguring the nuts and bolts of the database that is doing the computation or holding the data. And we are also reconfiguring the type of the virtual machine that is being given to you, right? So for example, Amazon has about 150 different VM types, and some are much more expensive and some are less expensive. So if you're unnecessarily getting a much more expensive VM, you would be paying a much higher cloud bill. So we right-size the type of the machine that is used for the cloud computation. So we are doing this blazing fast computation, but being mindful of the cost that you're going to pay at the end. We also have two kinds. So if you guys, if you have people who want to look into the latest bells and whistles of cloud computing, it's called serverless cloud computing. So last year we won a best paper award on how do you do this for serverless cloud computing. So I'm not going to go into the details of serverless, but I encourage you to go look at our paper called Wise Views, where we are able to do this for serverless cloud computing as opposed to the conventional cloud computing as well. So if nothing else, you can go and Google serverless cloud computing and you can see. Basically, I like thinking of serverless as the Ubers of the world, right? So you have rental cars, that is the conventional cloud computing, and serverless is pay as you go. So you pay exactly the amount of compute you're using rather than for the entire amount of time that you're renting the computer. So this is all published. I was mindful to not put any unpublished work. So all of this is available on my website under publications, including a best paper award in a computer performance. It's called Sigmetrics from last year. So feel free to look at us. And now I'm going to do this huge jump to go to the last slide because I don't think I want to stuff much more, but I do want to thank my student who recently graduated and now works in Intel and was also a postdoc in my lab for about a year, who did a lot of this optimization. Again, this is based on machine learning. How do you do the optimization for blazing fast cloud computation? And you can also do computer vision algorithms on the cloud. And you can also adapt to different kinds of latency and cost targets, again, which is a common theme related to the computer vision stuff. I'm going to directly skip to the overall takeaways. So I talked about a bunch of different things. One is how do you cluster and interpret single cell genomics data? Hopefully you remember the autoencoder part. So if you remember, I was making a photocopy. So you have the encoding or the embedding in the latent space and the decoding that you're doing. If that decoder gives you kind of similar to what you had originally, as an almost a photocopy, that means your latent space embedding is good, right? Depending on how much difference, how much drift is there between the original data and the decoded data. So can we deal with noise? So I told you, you have batch correction. We are able to deal with batch to batch variation. We are able to deal with instrument dropout events that result from some of these instrumentation. And we are also able to deal with matrices with lots of zeros, right? And we neuralize the clustering procedure. We use something called k-means clustering. We've also been using better and more powerful clustering such as Louvain and Leiden clustering. And we're also able to give you what are the important gene panels that typify these different clusters, right? So on the left, you see what we have clustered, and on the right, you see the competitor algorithm. As you can see, we are able to cluster much better, and we are able to come up with these interpretability of these clusters much better. So this work is available here if you're interested. I kind of went super fast talking about cloud computing for complex data analytics, because that is the data engineering part of my lab, where we are also using machine learning. We are using machine learning to save the earth from the big data gas guzzlers that these big companies have, right? So how can you use the right amount of computation for your machine learning? And then also being able to approximate computing. So think of being able to do computer vision on wearables on small devices or surgical robots, etc. So you basically not only have the ability to downsize your computation, but you're paying less in terms of less energy, using less energy, and giving more timely results. With that, I kind of gave you a plug for my company. We are hiring. So if you think you care about machine learning, you are good with Python and some of these programming languages, you can go look up KeyByte jobs. We're hiring 3 interns for summer. I have 1 spot remaining. And I'm not going to go into this. I will directly go, our product is Optimus Cloud, and you can look us up in keybyte.xyz. I have to skip through this as well and go to the slide. So College of Engineering has been profiling researchers where you have, different blogs to kind of explain some of the work. So I encourage you to go and look this up. Some of our genomics algorithms can be used to decrease the off-targeting for mRNA-based therapeutics. We're also bringing the cloud back to the earth in the sense that if you want to do a lot of this computation on your device, that's what this shows. And then we're taming the wild streaming algorithms that take a lot of compute, right? So how do you basically trim them down so you can run them on these small devices? So we are fast, we are furious, and we are interpretable. With that, this is all my funding, and this is my growth. Thank you.
Thank you for this fantastic talk, Professor Chaterji. Several questions came in. So one of them is, what is your insight regarding the complexity of algorithm, data set size, dimensionality, feature selection versus predictive power? Is complex better towards prediction or not? For example, it has been found that in some instances, machine learning or AI-based prediction do not outperform conventional regression analysis.
Wow, that's a very powerful question. So I'll try to remember all the different facets of the question, but I kind of got the gist of it, right? So yeah, I feel like oftentimes I teach 2 machine learning courses. I teach IoT and computer vision, and I teach computational genomics to undergrads. So I think oftentimes people just want to throw neural networks at everything. You don't have to do that. Sometimes a simple regression works very well, right? And sometimes simplicity is important. So if you look at actually a paper from when I was doing my postdoc in computer science, we had this paper where we saw that there is this thing called bias variance trade-off. So whoever asked this question, you can think of bias, like if an algorithm is underfitting, right? You see that, hey, you have this training versus test data, right? So if it is underfitting on the test data, you basically need a more powerful algorithm, and only then do you move to a more powerful algorithm. So don't always throw the most complex algorithm, because 1, you'll need more compute, and 2, your algorithm will not be as easy to interpret, right? So don't always use a neural network where you can use a simple regression, linear regressor, or what have you.
Thank you. Another question is for Kratos. Did you try different numbers of hidden layers before you decided to use 3 layers? If so, did you observe any effect of increasing, decreasing hidden layers on the model performance?
Yeah, nice question. So we used a very simple 3-layer neural network, right? So we just have, I think in this specific one, the final algorithm, we have 2 hidden layers. But yeah, if you use more number of hidden layers, you'll get potentially higher performance, and you can increase the number of hidden layers. But our proof of concept is, just use a simple neural network. Because remember, at the end of the day, you also care about 1, we wanted to have less number of parameters. We also compared the runtime. So the more complex the algorithm, the more runtime it will take, and the less interpretable it will be. But yeah, you can possibly squeeze in more performance by just adding more number of hidden layers.
Thank you. For the encoder-decoder component you discussed, what was the loss function used to optimize the algorithm?
Yeah, so for the encoder-decoder, typically what is used is the reconstruction loss. So remember I said if you have this photocopy, so the decoder is finally giving you this copy. So that reconstruction loss tells you how good your embedding is. So we use the reconstruction loss on one end, and the other loss was KL divergence. So we have the regular loss function, the cross entropy, or the KL divergence, and then we also have the reconstruction loss from the encoder-decoder.
Thank you. For assessment of model performance of Kratos, since you mentioned class imbalance, did you look at performance metrics other than AI ROC, such as precision recall curve?
Oh, nice. So for example, if you remember, I said AMI handles class imbalance a little bit better. That's how we had class imbalance in addition, that's how we had AMI in addition to ARI. But you're right, we can use a precision recall curve, and precision recall curve would be a nice way to handle class imbalance better. So if you see some of our past papers, for example, we have Avishkar, and I think we have a paper in TCBB from 2019 or 2020, and we have a nice comparison for it. You're right, if you have class imbalance, PR curves would be a nice tool, but yeah. You get only this many, so when you submit to a conference, you're limited to, based on the conference, 10 to 15 pages, so we have to be mindful of the final page budget as well.
Thank you. Thank you for your awesome talk. How can you check that this represents single-cell genomics accurately? And is your code user-friendly?
Yeah, that you have to ask Zihan, but as far as I know, it is user-friendly because it was also evaluated. So yeah, I would very much like for you to go and try out our code. And this single-cell genomics annotation is an active area in our lab, so we are doing a lot more with it. But yeah, we evaluated against what's considered the best. I think ACE had the best results before us. And our code is open source, so you can go try our code. And if you have questions, Zihan is the developer, but yeah, feel free to come back to us if you have questions.
What cell profiles and what kinds of data set did you use for the input model?
Yeah, so that's where, so we have another paper under submission. I didn't talk much about that because that is unpublished. So one of the things that people do is they increase the complexity of the data set, right? So I gave you some aspect of that, where you have the PBMC data set, which is a blood cell type mononuclear. It's from those two citations, and they have thankfully made their data outsourced. And a lot of these other baseline algorithms use these two data sets, so they are widely used. And these are called like benchmark data sets, so that you can compare apples to apples, right? You can compare on the same data sets. So we use the PBMC data set, and I think the Barron data set is the more complex data set is a pancreatic data set, but we've been using even more complex data sets. So one of the things that we've seen is for the future, for the current version of our algorithm that we are working on, if you have more complex data sets, because of the kinds of embeddings that we are generating, we are able to do even better, like relatively even better than the baselines on more complex data sets. So the baselines suffer a lot on the complex data sets, whereas we are able to do really well. The other thing is that SimSim package. The SimSim package lets you simulate data sets with varying degrees of complexity so that you can also stress test your algorithm by trying out different kinds of complexity by simulating these data sets in addition to real data sets.
Thank you. Another question is 500 cells enough? For the RNA-seq analysis, we always expect that at least 1,000 cells for the next step analysis.
Yeah, good point. Yeah, so we are increasing the data set complexity. As you know, a lot of these data sets that are used as benchmark data sets have to go through some, rigorous standards. They should be, well tested and everything. So the current data sets that we are using are even larger. But yeah, these are, I think the one you mentioned is the PBMC data set, but the Barron data set has a higher number of cells. And now we are using Plastrid, PLASSCHAERT. You can look that up. They have even higher number of cells. So once you get, more and more data sets, because we don't generate these data sets. These are outsourced by experimental labs that generate these data sets. So yeah, our current data sets have higher number of cells. And like I mentioned, even on these more complex large data sets, we fare really well because of the kinds of embeddings that we are generating.
It's a generic question. From your viewpoint, what are some major upcoming problems in the field of machine learning?
Yeah, well, yeah, this is almost like, at least from my standpoint, like the projects that we are funded on, and obviously the proposals that are are what I want to work on. So I'm always caught in two minds when I think of, one is we are an applied machine learning lab. So oftentimes what gives me a lot of joy is we can apply the same kinds of algorithms to completely different domains. So right now, what we work on is a lot of the single cell genomics data sets, because that's what a lot of data is coming out from the single cell instrumentation. But there are lots of problems, sort of bottlenecks on the analysis side. So we can use these latent embeddings to come up with these denoised embeddings. And on the computer vision side as well, you have, think of all these self-driving cars, right? They need to run these algorithms real time on these small mobile GPUs. So a lot of these approximate algorithms, what I'm really excited about is being able to approximate these algorithms so that they can run on these small devices, and they can also run in an energy-aware manner, right? Because if you run really unwieldy, large neural networks, they also will need really large GPU computers. We also use those. We have a lot of those in our lab, but we now have like about 25 students. Everyone's kind of making, during a deadline, people are trying to use the same GPU machines. So if you can, not only from the standpoint of deadlines, but if you can use these algorithms on, less resourced computers, you can run them one in an interpretable manner, two in a more energy efficient manner. So I feel like approximating algorithms, being able to run them on simple devices, and being able to interpret these algorithms for application scientists, right? Like I said, thinking of the clusters, thinking of the gene enrichment in these clusters, right? So not just coming up with an output of the machine learning algorithm, but can you make sense of this using methods that are amenable or palatable to the application scientists? And then go back and say, if I find a new cell type, can I say, hey, can you guys go and see what are the kinds of genes that are enriched in this cell type? And maybe you'll find that, hey, this specific kind of Alzheimer's state causes an enrichment in that specific cell type that was not known before. So being able to come up with these new predictions that have not been known before is also very exciting. And for that, you need to have your algorithms interpretable.
Well, thank you so much. That's all the questions. Thank you for your time for this fantastic talk, Professor Chaterji.
Yeah, thank you. This was a lot of fun. Take care.
Very nice.
Bye.